question is that, what is biological warfare? Welcoming everyone, I am starting another new episode of World Tribune. I am Nadim Mahmoud Nayan with you. If you are new to our channel, please stay with us by pressing the subscribe or follow button, and if you have already subscribed, thank you. So let's move on to our original report. According to Wikipedia, biological warfare, also known as germ warfare, is the use of biological toxins or infectious agents such as bacteria, viruses, insects, and fungi with the intent to kill, harm or incapacitate humans, animals or plants as an act of war. Biological weapons, often termed bioweapons, biological threat agents, or bioagents, are living organisms or replicating entities, i.e., viruses, which are not universally considered alive. Entomological, insect warfare is a subtype of biological warfare. Offensive biological warfare is prohibited under customary international humanitarian law and several international treaties. In particular, the 1972 Biological Weapons Convention, BWC, bans the development, production, acquisition, transfer, stockpiling and use of biological weapons. Therefore, the use of biological agents in armed conflict is a war crime. In contrast, defensive biological research for prophylactic, protective or other peaceful purposes is not prohibited by the BWC. Biological warfare is distinct from warfare involving other types of weapons of mass destruction, WMD, including nuclear warfare, chemical warfare, and radiological warfare. None of these are considered conventional weapons, which are deployed primarily for their explosive, kinetic, or incendiary potential. Biological weapons may be employed in various ways to gain a strategic or tactical advantage over the enemy, either by threats or by actual deployments. Like some chemical weapons, biological weapons may also be useful as area denial weapons. These agents may be lethal or non-lethal, and may be targeted against a single individual, a group of people, or even an entire population. They may be developed, acquired, stockpiled or deployed by nation-states or by non-national groups. In the latter case, or if a nation-state uses it clandestinely, it may also be considered bioterrorism. Biological warfare and chemical warfare overlap to an extent, as the use of toxins produced by some living organisms is considered under the provisions of both the BWC and the Chemical Weapons Convention. Toxins and psychochemical weapons are often referred to as mid-spectrum agents. Unlike bioweapons, these mid-spectrum agents do not reproduce in their host and are typically characterized by shorter incubation periods. Overview. A biological attack could conceivably result in large numbers of civilian casualties and cause severe disruption to economic and societal infrastructure. A nation or group that can pose a credible threat of mass casualty has the ability to alter the terms under which other nations or groups interact with it. When indexed to weapon mass and cost of development and storage, biological weapons possess destructive potential and loss of life far in excess of nuclear, chemical, or conventional weapons. Accordingly, biological agents are potentially useful as strategic deterrents, in addition to their utility as offensive weapons on the battlefield. As a tactical weapon for military use, a significant problem with biological warfare is that it would take days to be effective, and therefore might not immediately stop an opposing force. Some biological agents, smallpox, pneumonic plague, have the capability of person-to-person transmission via aerosolized respiratory droplets. This feature can be undesirable, as the agents may be transmitted by this mechanism to unintended populations, including neutral or even friendly forces. Worse still, such a weapon could escape the laboratory where it was developed, even if there was no intent to use it, for example by infecting a researcher who then transmits it to the outside world before realizing that they were infected. Several cases are known of researchers becoming infected and dying of Ebola, which they had been working with in the lab, though nobody else was infected in those cases. While there is no evidence that their work was directed towards biological warfare, it demonstrates the potential for accidental infection even of careful researchers fully aware of the dangers. While containment of biological warfare is less of a concern for certain criminal or terrorist organizations, it remains a significant concern for the military and civilian populations of virtually all nations. Antiquity and Middle Ages. Rudimentary forms of biological warfare have been practiced since antiquity. The earliest documented incident of the intention to use biological weapons is recorded in Hittite texts of 1500 to 1200 BCE, in which victims of tularemia were driven into enemy lands, causing an epidemic. The Assyrians poisoned enemy wells with the fungus ergot, though with unknown results. Scythian archers dipped their arrows and Roman soldiers their swords into excrements and cadavers. Victims were commonly infected by tetanus as a result. In 1346, the bodies of Mongol warriors of the Golden Horde who had died of plague were thrown over the walls of the besieged Crimean city of Kaffa. Specialists disagree about whether this operation was responsible for the spread of the Black Death into Europe, Near East and North Africa, resulting in the deaths of approximately 25 million Europeans. Biological agents were extensively used in many parts of Africa from the 16th century AD, most of the time in the form of poisoned arrows, or powder spread on the war front, as well as poisoning of horses and water supply of the enemy forces. 1920, in Borgu, there were specific mixtures to kill, hypnotize, make the enemy bold, and to act as an antidote against the poison of the enemy as well. The creation of biologicals was reserved for a specific and professional class of medicine men. International law. International restrictions on biological warfare began with the 1925 Geneva Protocol, which prohibits the use but not the possession or development of biological and chemical weapons. Upon ratification of the Geneva Protocol, several countries made reservations regarding its applicability and use in retaliation. Due to these reservations, it was in practice a no first use agreement only. The 1972 Biological Weapons Convention, BWC, supplements the Geneva Protocol by prohibiting the development, production, acquisition, transfer, stockpiling and use of biological weapons. Having entered into force on the 26th of March 1975, the BWC was the first multilateral disarmament treaty to ban the production of an entire category of weapons of mass destruction. As of March 2021, 183 states have become party to the treaty. The BWC is considered to have established a strong global norm against biological weapons, which is reflected in the treaty's preamble, stating that the use of biological weapons would be repugnant to the conscience of mankind. The BWC's effectiveness has been limited due to insufficient institutional support and the absence of any formal verification regime to monitor compliance. In 1985, the Australia Group was established, a multilateral export control regime of 43 countries aiming to prevent the proliferation of chemical and biological weapons. In 2004, the United Nations Security Council passed Resolution 1540, which obligates all UN member states to develop and enforce appropriate legal and regulatory measures against the proliferation of chemical, biological, radiological, and nuclear weapons and their means of delivery, in particular, to prevent the spread of weapons of mass destruction to non-state actors. Bioterrorism. Biological weapons are difficult to detect, economical and easy to use, making them appealing to terrorists. The cost of a biological weapon is estimated to be about 0.05% the cost of a conventional weapon in order to produce similar numbers of mass casualties per kilometer square. Moreover, their production is very easy as common technology can be used to produce biological warfare, like that used in production of vaccines, foods, spray devices, beverages and antibiotics. A major factor in biological warfare that attracts terrorists is that they can easily escape before the government agencies or secret agencies have even started their investigation. This is because the potential organism has an incubation period of 3 to 7 days, after which the results begin to appear, thereby giving terrorists a lead. A technique called clustered, regularly interspaced, short palindromic repeat, CRISPR-Cas9, is now so cheap and widely available that scientists fear that the amateurs will start experimenting with them. In this technique, a DNA sequence is cut off and replaced with a new sequence or code that codes for a particular protein or characteristic, which could potentially show up in the required organism. Though this technique is a breakthrough and is commendable, it can cause serious issues and potential danger if used by people with wrong intentions. Concerns have emerged regarding do-it-yourself biology research organizations due to their associated risk that a rogue amateur DIY researcher could attempt to develop dangerous bioweapons using genome editing technology. In 2002, when CNN went through Al-Qaeda's, AQ's, experiments with crude poisons, they found out that AQ had begun planning ricin and cyanide attacks with the help of a loose association of terrorist cells. The associates had infiltrated many countries like Turkey, Italy, Spain, France and others. In 2015, to combat the threat of bioterrorism, a national blueprint for biodefense was issued by the Blue Ribbon Study Panel on Biodefense. Also, 233 potential exposures of select biological agents outside of the primary barriers of the biocontainment in the U.S. were described by the annual report of the Federal Select Agent Program. Though a verification system can reduce bioterrorism, an employee, or a lone terrorist having adequate knowledge of the company facilities, can cause potential danger by injecting a deadly or harmful substance into the facility. Moreover, it has been found that about 95% of accidents that have occurred due to low security have been done by employees or those who had a security clearance. Entomology. Entomological warfare, EU, is a type of biological warfare that uses insects to attack the enemy. The concept has existed for centuries and research and development have continued into the modern era. EU has been used in battle by Japan, and several other nations have developed and been accused of using an entomological warfare program. EU may employ insects in a direct attack or as vectors to deliver a biological agent, such as plague. Essentially, EU exists in three varieties. One type of EU involves infecting insects with a pathogen and then dispersing the insects over target areas. The insects then act as a vector, infecting any person or animal they might bite. Another type of EU is a direct insect attack against crops. The insect may not be infected with any pathogen but instead represents a threat to agriculture. The final method uses uninfected insects, such as bees or wasps, to directly attack the enemy. Genetics. Theoretically, novel approaches in biotechnology, such as synthetic biology, could be used in the future to design novel types of biological warfare agents. Would demonstrate how to render a vaccine ineffective. Would confer resistance to therapeutically useful antibiotics or antiviral agents. Would enhance the virulence of a pathogen or render a non-pathogen virulent. Would increase the transmissibility of a pathogen. Would alter the host range of a pathogen. Would enable the evasion of diagnostic detection tools. Would enable the weaponization of a biological agent or toxin. Anti-personnel. Ideal characteristics of a biological agent to be used as a weapon against humans are high infectivity, high virulence, non-availability of vaccines, and availability of an effective and efficient delivery system. Stability of the weaponized agent. The ability of the agent to retain its infectivity and virulence after a prolonged period of storage may also be desirable, particularly for military applications, and the ease of creating one is often considered. Control of the spread of the agent may be another desired characteristic. The primary difficulty is not the production of the biological agent, as many biological agents used in weapons can be manufactured relatively quickly, cheaply, and easily. Rather, it is the weaponization, storage, and delivery in an effective vehicle to a vulnerable target that pose significant problems. Anti-crop, anti-vegetation, anti-fisheries. The United States developed an anti-crop capability during the Cold War that used plant diseases, bioherbicides, or mycoherbicides, for destroying enemy agriculture. Biological weapons also target fisheries as well as water-based vegetation. It was believed that the destruction of enemy agriculture on a strategic scale could thwart Sino-Soviet aggression in a general war. Diseases such as wheat blast and rice blast were weaponized in aerial spray tanks and cluster bombs for delivery to enemy watersheds in agricultural regions to initiate epiphytotic epidemics among plants. On the other hand, some sources report that these agents were stockpiled but never weaponized. When the United States renounced its offensive biological warfare program in 1969 and 1970, the vast majority of its biological arsenal was composed of these plant diseases. Enterotoxins and mycotoxins were not affected by Nixon's order. Thank you so much for watching our report. To get such informative videos, subscribe to our World Tribune YouTube channel and follow the Facebook page. See you again in a new episode with a new topic. Stay well then stay healthy.